Today, we will finish the discussions about positive and negative controls. This will be part 2 of a summary of control experiments. And we will also conclude step 11 for reading the Milestone Article 1, the most important paper written by the Nobel Laureates of 2023. We will talk about the 3 roles that positive and negative controls play in life science experiments and papers. The examples will be taken from the Milestone Article 1. If we do not understand the 3 roles, the data with some positive controls might appear strange and confusing. But if we do understand the 3 roles, we will understand the data clearly, and we will understand the story of the paper clearly. So far, we have discussed positive and negative controls in detail in many episodes. They could be a little too advanced for those who just want to read and remember textbooks, or for those who just want to read published papers and passively accept what the authors say. But if you have enjoyed following these episodes, it means that you are interested in fine details. I'm sure you will be a careful person, and you will grow your capability to perform experiments critically and read papers critically. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologica. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto: learn the skills, enjoy academic life. Previous episodes. We have been discussing how to read life science papers using one of the best examples. The paper is the Milestone Article 1, published in 2005 by the laureates of the Nobel Prize in Physiology or Medicine 2023. The laureates were Dr. Katalin Karikó and Dr. Drew Weissman. I have added the links to the paper in the episode description. If you are a new listener of my episodes, please start listening from episode 7. It was the start of the mini-series of reading. The last episode of the mini-series of reading was part 1 of a summary of control experiments. We discussed that the positive controls in Milestone Article 1 reported variable values when the cell types were different, or even when the same cell type was used. Our conclusion was that it is important to include both positive and negative controls in the same experimental run. Today's episode. First, we will review and further discuss how the positive and negative controls were used in Milestone Article 1. This will be part 2 of a 2-part summary of control experiments. Second, we will conclude Step 11 for reading the Milestone Article 1. Step 11 is for examining the milestone graph. In the next step, we will start reading the text of the paper. 3 roles of positive and negative controls. In our episodes, we have discussed at least the following 3 roles of the positive and negative controls so far. All of them are helpful for us from the following 2 perspectives. 1, experimenter's perspective when we do experiments, and 2, reader's perspective when we read papers and their figures. The first role of positive and negative controls, health checkup of the experimental system. The first role of positive and negative controls is to help us determine if the experimental system is operating as intended and it is free from errors. There are at least 4 cases when the controls play this first role. In the first case, the controls are important for checking the experimental system when we set up new experiments and when we find a problem and troubleshoot it. We specifically talked about using negative controls for troubleshooting. That was in 7 episodes from the episode number 19 through number 26. We discussed the story about a pilot experiment that failed at first but was made successful after troubleshooting. Here are the keywords as reminders: the negative controls and the serum in culture medium. In the second case, the controls are important for checking the experimental system when we train a new person in the lab to do experiments. When a new person does an experiment, we will definitely need to include positive and negative controls in the experiment. We, as mentors, will look at the results of the positive and negative controls, and we can judge whether the new person followed the instructions carefully and correctly, and whether the person had good hands for doing the experiments. The good hands mean both the manual and mental dexterities in the lab. In the third case, the controls are important for checking the experimental system when we read a paper. We will check if the experiments are working well by examining how well the positive and negative controls in the figures work as expected. As the fourth case, let's review practical examples in Milestone Article 1. They were the positive controls for the toll-like receptors and the mnemonic that we created. The laureates characterized the synthetic chemicals poly-IC and R848 as positive controls for specific toll-like receptor subtypes. For this purpose, they used the HEK293 model cells. This was reported early in the paper, that is, in figure 2b, B as in Bravo, out of 6 main figures plus 1 supplementary figure. The laureates showed that poly-IC selectively activates the toll-like receptor 3, and R848 selectively activates toll-like receptors 7 and 8. We created a mnemonic for easy reading. The mnemonic was poly-IC3 R84878. Based on this figure 2b, we can interpret poly-IC as a positive control for toll-like receptor 3 and R848 as a positive control for toll-like receptors 7 and 8. This finding was consistent with what was reported in the literature. We discussed these in episode 29. Side note. Please note that our mnemonic will work only for the purpose of reading the Milestone Article 1. If you are working in the immunology field, please be very careful when you use this mnemonic. It is possible that these compounds are now known to work in an additional way or in another way based on the work published after the Milestone Article 1. So our mnemonic could be too simplistic, and it could be ignoring subtle nuances about how these compounds should be used for. It is possible that the mnemonic could be inaccurate now. But for the moment, it will help us while we read the Milestone Article 1. Back to the main content. The second role of positive and negative controls, references of comparison. The second role of positive and negative controls is to serve as references of comparison so that we can interpret the measured values of the test group that would typically fall between the positive and negative controls. There are 2 broad cases. In many cases, the positive controls indicate the upper limit of the measured values, and the negative controls indicate the lower limit of the measured values. But in some cases, the relationship could be the opposite. For example, if the experiment tests the effect of inhibiting a specific phenomenon, a positive control will inhibit maximally. Then, the positive control will show the lower limit of the measured values of the phenomenon, and the negative control will indicate the upper limit of the measured values. In either case, the positive and negative controls show the range of possible measured values. So they serve as references for interpreting the measured values of test groups that would typically fall somewhere within the range. One caution here. When the experimental condition changes, the measured values of the positive and negative controls could change too. Sometimes the measured values could change drastically. So each experiment needs to include positive and negative controls. We already talked about good examples in our Milestone Article 1. In the episode 32, we saw that the same positive control, R848, can stimulate the three types of dendritic cells to different degrees. There was a large difference in the concentrations of the released cytokine, tumor necrosis factor alpha. Because of this fact, we learned again that every experiment needs both positive and negative controls. Here are the three types of dendritic cells used in the work. The first cell type was the primary dendritic cells labeled as DC1 and DC2. The second cell type was the monocyte-derived dendritic cells, abbreviated as MDDC. And the third cell type was the interferon alpha-generated monocyte-derived dendritic cells, abbreviated as IFN alpha MDDC. They all belong to the dendritic cells, but the behaviors were very different. Summarizing the first two roles of positive and negative controls, we can say that by including both controls in an experiment, we can examine if the experiment is working well as expected, and we can interpret the measured values of the test groups using the controls as references. So the positive and negative controls determine the experimental designs, and they also strengthen the interpretations of the experimental results. The third role of positive and negative controls, characterization of experimental systems. The third role of positive and negative controls is to characterize new experimental systems. In an earlier part of a paper, the authors can specify positive and negative controls and use them in their first and second roles, that is, showing that the experimental system works well and showing a range of measurements. Then later in the same paper, when the experimental conditions change, the authors can use the same controls for characterizing the changed experimental system. In this case, the previously used controls might not necessarily show the upper and lower limits of the measured values. We briefly talked about an example in our Milestone Article 1. We discussed it in episode 30. In this example, positive controls served as indicators of expressions of specific toll-like receptor subtype among different cells. In Figure 2, poly-IC was identified as a positive control for toll-like receptor 3, and R848 was identified as a positive control for toll-like receptors 7 and 8. In the next figure, Figure 3D, D as in delta, the following two facts were shown. 1. A positive control, R848, reliably stimulated the cytokine release in one of the primary dendritic cells, DC1. 2. But another positive control, poly-IC, did not stimulate cytokine release in the same cells. And the interpretation was that poly-IC can be thought of as an indicator of whether the toll-like receptor 3 was expressed in the tested cells. So we can also learn that poly-IC did not work as a positive control in all experiments in the paper. This situation was rather simple. In the next section, you will see that there were more complex situations. More about the third role of positive and negative controls. We would like to elaborate on this third role of positive and negative controls in our Milestone Article 1. Specifically, we will examine more examples of how the positive controls served as indicators of expression of specific subtypes of toll-like receptors. The key point was how much of the same cytokine, tumor necrosis factor alpha, was released by poly-IC in comparison to R848. So we will look at the relative values in each experiment. We already talked about the results using the primary dendritic cells. We found that in Figure 3D, left panel, poly-IC value was almost 0, but the R848 value was substantial. In the next 5 examples, taken from 5 figure panels, a different cell type was used. The cells were monocyte-derived dendritic cells, abbreviated as MDDC. You will see 4 types of variations among the 5 figure panels. We will talk about the variations starting from the low poly-IC value relative to the R848 value. In Figure 1 and supplementary figure S1A, poly-IC value was about half of R848 value. In Figure 3C, poly-IC value was about 2/3 of R848 value. In Figure 4B, poly-IC value was almost the same as R848 value. And in Figure 3A, left panel, poly-IC value was almost 2-fold higher than R848 value. So among the 5 figure panels, the poly-IC value varied from about half to 2-fold of the R848 value. And among the 6 figure panels, including the data with primary dendritic cells, the poly-IC value varied from about 0 to 2-fold higher than the R848 value. What did the laureates say about these very wide variations? In the 7th paragraph of the results section, the laureates mentioned a few of these figure panels. Interestingly, the laureates indicated that the monocyte-derived dendritic cells from different donors showed different responses to the 2 positive controls. To illustrate more variations, I added more examples here to make a systematic comparison between poly-IC and R848. These variations can be interpreted this way. Monocyte-derived dendritic cells in different experiments expressed different levels of toll-like receptors. In the cells of some experiments, the expression of toll-like receptor 3 was lower than the expression of toll-like receptors 7 and 8. In the cells of other experiments, the relationship was the opposite. The expression of toll-like receptor 3 was higher than the expression of toll-like receptors 7 and 8. Such differences in the relative receptor expressions could lead to variable responses to poly-IC and R848. Quick note: we just interpreted the variable responses to poly-IC and R848 as representing variable expressions of toll-like receptors 3, 7, and 8. Here, it is important that we are assuming we can apply our mnemonic to the immunological dendritic cells. But please note that the results that led to our mnemonic were obtained in the HEK293 model cells. Our interpretation would be correct if the mechanisms in the HEK293 model cells are the same as in the dendritic cells. But our interpretation could be incorrect if the mechanisms are different in different cell types. So we will need to be careful when we interpret data obtained under different experimental conditions. In this case, the data were obtained from different types of cells. But in our discussion about the third role of positive controls, the fact will still remain the same. There were variable responses to the 2 positive controls, poly-IC and R848. So our discussion is still good. Now back to the main content. Summary of the 3 roles of positive and negative controls. The first role of positive and negative controls was to help us determine if the experimental system is operating as intended and it is free from errors. The second role of controls was to serve as references of comparison, showing the upper and lower limits of measured values. And the third role of controls was to characterize new experimental systems. As for the first role as a checker of the experimental system, we discussed 4 cases. When we troubleshoot the problems associated with the experimental system, when we train a new person in the lab, when we read a paper, and when we confirm what is reported in the literature. As for the second and third roles, as serving as references of comparison and characterizing new experimental systems, we used the two synthetic compounds, poly-IC and R848, as examples of positive controls, and they were applied to the natural immune cells, that is, the dendritic cells. We can say that those two compounds are very important because they were used in almost all figures, that is, in figures 1 through 4 and figure 6 and one supplementary figure. They were not used in the only remaining figure 5. In those figures, we have seen that R848 stimulated the cytokine release relatively constantly in multiple experiments. In contrast, poly-IC stimulated very variably, ranging from the almost zero level to twofold higher level than R848. The two synthetic compounds stimulated the cytokine releases to variable degrees. They did not always report the upper limits of the measured values, so they did not play the second role of positive controls, that is, the references of comparison. Based on these discussions, we will have one question naturally. Do these discussions mean that there were no true positive controls in those experiments? The answer is that there were true positive controls. The goal of those main experiments was to answer one major question. If RNAs contain modified nucleosides, will they suppress the cytokine release from the dendritic cells in comparison to the RNAs with unmodified nucleosides? To answer this question, the laureates compared the effect of the modified forms of in vitro transcribed RNA with the effect of the unmodified forms. So the unmodified forms served as positive controls, and they were included in almost all the figures. Based on all these discussions, I would say that there are unhelpful thinking and helpful thinking about positive controls. The unhelpful thinking is that poly-IC and R848 always serve as positive controls for immune responses, always reporting the largest measurement values throughout the paper. This statement was true in the HEK293 model cells, but in native immune cells, like dendritic cells, the positive control poly-IC even reported a value as low as that of a negative control. So it would be misleading to identify poly-IC and R848 always as positive controls throughout a paper. Instead, the helpful thinking is that 1. The terms positive and negative controls could be blurred and nuanced in experiments under new conditions or under new experimental designs in the same paper. And 2. We will need to carefully interpret which conditions will serve as positive and negative controls in each experiment. In this context, the laureates were wise. As I mentioned before in episode 28, the laureates were careful not to use the full terms positive control or negative control in any part of Milestone Article 1. After all these discussions, I hope you agree that it was good for us to change our mindset about positive and negative controls by modifying their definitions. One of the standard definitions would be to define them as experimental conditions under which a response is known to be present or absent. But we modified them. They are defined as experimental conditions under which a response can be thought to be present or absent. The modified definitions will help us become careful, cautious, and flexible when we design experiments, when we interpret experimental results, and when we read papers. Our Milestone Article 1 showed us multiple roles of positive and negative controls and how the controls could behave differently under various conditions. We learned a lot from one of the best papers in life sciences. This is the end of the discussions about positive and negative controls. By this, we finished step 11-6 of reading the Milestone Article 1. This is also the end of step 11. Let's briefly summarize step 11 here. Summary of entire step 11. After quickly navigating the figures in step 10, we moved to step 11. The goal of step 11 was to understand the milestone graph. This graph, or a figure panel, was shown specifically in the presentation video by the Nobel Committee. This graph is the leftmost graph in figure 3D, D as in delta, and it is the most important graph in our Milestone Article 1. We examined this graph and its related figure panels in detail. Step 11 had 6 sub-steps. In step 11-1, we interpreted the basic elements of the graph by identifying the horizontal and vertical axes of the bar graph. In step 11-2, we looked for the explanations of the graph in the figure legend. In step 11-3, we deciphered the labels in the graph, that is, any text elements that could help us understand the graph. In step 11-4, we understood the basic experimental conditions by reading the axis labels. In the specific examples in this article, the category axis labels were structured in a beautiful manner so that the labels indicate the experimental conditions visually and clearly. In step 11-5, we identified the negative control in the milestone graph. And in step 11-6, we identified the positive controls of experiments. This step 11-6 is the last part of step 11, and we concluded it today. Steps 11-5 and 6 showed the advanced viewpoints for negative and positive controls. We went well beyond the minimum knowledge. This experience will help you read the paper at a deep level. Why step 11 is possible. In step 11, we examined the milestone graph in figure 3D in detail and in depth. Step 11 was possible because we have already watched the beautiful presentation video by the Nobel Committee. The presentation part of the video is only 9 minutes long, and it is easy to understand. I will show the link to the video in the episode description. The presentation showed us why the laureates did the specific experiment of the figure, how the laureates did the experiment, what this figure shows, and what the result means. The Nobel Committee prepared very helpful illustrative diagrams for us too. In other words, step 1 was possible because we have already learned the overview of the work using the milestone graph as a guide. Why step 11 is necessary. In step 11, we carefully examined the details of the milestone graph, which forms the core data underlying the major conclusions of the work. The milestone graph will drive home the main message of the paper in a graphical format, even before we read the article. Step 11 is necessary because examination of the core data will help us understand the core message, and the understanding of the core message will prepare us for reading the text of the paper in the next steps. Let's wrap up. Summary. Today, we finished talking about the positive and negative controls. We focused on their 3 roles. The first role was to help us determine if the experimental system is operating as intended and it is free from errors. The second role was to serve as references of comparison, showing the upper and lower limits of measured values. And the third role of positive and negative controls was to characterize new experimental systems. We also discussed that these controls can play multiple roles in a single paper, sometimes in a confusing manner. Because of this, we think that our modified definitions of these controls will help us become careful, cautious, and flexible in our activities. For example, when we design experiments, when we interpret experimental results, and when we read papers. Then we concluded step 11, which is necessary for understanding the core message of the Milestone Article 1. This is it for today. We are now ready to start reading the text of the paper from the next step. Specifically, we will read the abstract in step 12. Your action. If you like this podcast, please share it with your friends, colleagues, mentors, or anybody who would be interested in it. This way, you and I can extend the reach. The bigger the audience becomes, the more feedback we can get, the better the episode quality will grow, and the more you and I can learn. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.